I'm going to try to give a really quick talk, but I was asked to talk about minimum maturity standards. And then I have one or two comments about what's up with rootstocks based on a request also to cover. So I'm going to cover minimum maturity standards. So if you're an avocado grower, we have minimum maturity standards in California for avocados. And this is just, the standards for California avocados is regulated by CDFA through a committee called Avocado Inspection Service. And they have an avocado inspection committee. The Avocado Inspection Committee does weights and measures, but it also regulates the enforcement of maturity standards. So this slide just shows that and the maturity standards for avocados, which I'll cover in the next couple slides, are fairly complicated, but we have other complicated maturity standards as well. And I think many of you grow citrus, and it's not only the sugar and acid component, it's also peel color. So there's other examples of, and in grapefruit, we actually have a double standard, one for the desert and one for everywhere else. So that is important here for the valley. So historical perspective on fruit maturity for avocados, in 1925, the standard was established using 8% oil. And then when we got to the 1970s, the components that were used for the analysis of the oil were deemed carcinogenic, and we needed to find a new quick and easy method, because when you're measuring minimum maturity, you need something that is really quick and inexpensive, because you have a lot of fruit you have to sample to make sure you meet the standard. So based on work that was conducted at UC Riverside that was funded actually by the California Avocado Commission, we changed to dry matter. So, and that work showed that there was a very high correlation between oil content and dry matter of the fruit. And then there was a group of growers and packers and other representatives in the industry, led by a group by Cliff Rainey, who followed up on the work of Lee. And you can see they did that work from 1988 to 1993. And the dry weight method is using a microwave oven, okay? The dry weight, and so we had dry weight standards established based on the work of Rainey and his group. And in 1995, though, they were slightly modified to what we currently have due to the request from Gwen growers. And I won't go into the gory details of that. Up until 2002, we used a method where you would take an a lengthwise slice of the fruit, and you would take it from multiple fruit, put it in a food processor, grind it up, and then microwave that ground tissue. That took, to prepare a sample for 5 fruit, which is a usual size of sample that is used, took 25 to 30 minutes plus the microwaving. So it was very laborious. You're working with a lot of knives. It was very dangerous. And in 2000 to 2002, myself working with Danella Borm from the Avocado Inspection Service and Ruben Hofsche, who was a packer and avid grower and innovator, we developed the coring method, because previous research has showed that if you look at the dry weight in the fruit, there's a gradient of the dry weight in the fruit where that from high to low. So the lowest dry matter is actually at the top of the seed, highest dry matter at the base of the seed, and actually the equator of fruit or in the middle of the seed is a very good approximation. It's not perfect, but a very good approximation of the average of the entire fruit. So by using the coring method, where you're just taking a sample, a coring machine was basically developed that is now used, and that was a regulation was changed in 2002, so that instead of taking 30 minutes to prepare 5 fruits, it takes 5 minutes. And no knives, or minimal knife work. Okay, so the California minimum maturity standard is a 2-step process. You have to have a minimum dry weight for each variety, and then we have a thing called release dates. And the genesis of the idea of release dates goes back to the research that Sukun Lee did back in the 1970s and 80s. This is just, we have many, every variety in California has a minimum dry matter. This is just some of the main varieties we grow in California, what that minimum dry matter is. These numbers are available on the California Avocado Commission website. And it's based on the work. Lee did most of the work. Rainey modified some of the work that Lee did. And then for good or bad, I'm responsible for the dry matter standards for lamb, Hass, and Gem. That all the dry matter standards were set by using taste panels or doing individual fruit assessment and relating it back to the dry matter. So it wasn't just a number pulled out of the air. There's actually some data and quite a bit of effort behind those dry matter numbers. Now, the other thing is the release dates. So the work that Lee did showed that actually you could use fruit size to sort of predict what the dry matter would be. And as a general rule, big fruit reached that minimum dry matter faster than small fruit. So we have this idea of release dates. So this is the release dates for Hass. So you can see if you have a size 48 fruit, which is the peak size, usually the peak size for Hass avocados, you can see the release date is December 12th. So what does that mean? If you want to go out and pick your Hass fruit before December 12th and you're size picking, you want to pick size 48, if you pick them before December 12th, you have to call the Avocado Inspection Service and say, okay, I'm picking today, and they need to verify that that fruit is the minimum maturity of 20.8 for Hass. If you pick on December 13th, you can pick whatever you want for that size. Okay, that's basically how the release dates work. So I know a lot of you are interested in growing Gem. Gem is a sort of a deviation from what I was just talking about. So Gem avocado is a variety of the University of California. It was a seed planted in 1986. It was evaluated and top worked into the research station in Irvine in 91, 92. And it was patented and released in 329.5. No one was interested in it, except for a very, very few people in Ventura County, and it just was a no-go. But however, in South Africa was interested in it. They became the master licensee of the Gem avocado. They started planting it in South Africa and elsewhere. And all of a sudden, in about 2014, I started getting calls from California growers saying, you gave away our material. What did you do? And I said, well, we released it in 2003. And it's been around, and I've been giving talks about it. And I had backup. I was able to demonstrate I had talked about it. But anyway, it's taking off, and it's a great variety. It's a smaller tree than Hass. Under proper management, it has less alternate bearing. And because it's a smaller tree, it potentially can plant it in high density. And more importantly, for here in the valley, it seems to be better adapted to high heat. It's not heat resistant, but it is more adapted to high heat. And we have data showing that the stomata closure in that high heat that Anon talked about occurs at a slightly higher temperature than Hass. So we established a minimum dry matter standard for Gem of 22.8. That was in 2006, but we didn't have any real acreage in California, and so it wasn't important to have release dates. But starting in 2018, we started doing works on release dates, and the release dates were established in 2022. So I just want to show you now what the basis of the release dates for Gem are. So you can see there's 3 years of data here. The red line is Riverside and San Diego and Orange counties. The blue line is Ventura and Santa Barbara counties. And in 2019 and 2020, you see the appearance of the green line. And that green line is Lynn Cove, as we're seeing here, Lemon Cove, and Reedley. So we were lucky enough to find some two growers that were willing to let me go and sample fruit off of there. And we sampled fruit every 2 weeks. So and you can see that the San Joaquin Valley data, when you average those three sites, and is much earlier, that reaches at 22.8 much earlier than it did in Southern California. So in fact, when you do the regression analysis, you can see that the proposed release date for a size 48 fruit was December 11th up here versus February 4th in Southern California. There was about a 10-day difference between the north and the south. And so that February 4th is that halfway point between that 10-day difference. So there was hearings from the Avocado Inspection Service. And here now, if you're a gem grower, you have a different set of release dates than the growers in Southern California. So this is the first time now in the maturity standards for California avocado that we have a split release date for varieties. If you're growing Hass or Lamb Hass, you're going to have to stick with the release dates that are already established for those varieties unless we conduct new additional research to show that we can have an earlier release date. But it doesn't mean you can't pick. You're just going to have the fruit tested and work with the Avocado Inspection Service. So I just want to make a couple of other comments to you based on, you'll see the trees at Lynn Cove. They haven't, we're not very good growers of avocados here at Lynn Cove. So you'll see maybe worst case. But I can tell you general observations. We have a companion trial in Irvine. The trees here tend to be a little bit larger. Is that because we have less yield? Or they just grow bigger here? I can't tell you. The architecture of the tree is different here than it is in Southern California. The yield is about 2/3 of what we can get in Irvine, but it is large. The difference is in fruit weight. It's not in the numbers of fruit that we pick. So I think we don't have overhead cooling on the trees, and maybe that would boost the yields. Like we saw with the release date data, the seasonality is enhanced, and the fruit colors up earlier than it does in Southern California. In fact, you won't see fruit on the trees today because we harvested 2 weeks ago because the Hass and the gem were starting to color up. And I know from the work that I've done in post-harvest that when you see color on the fruit, that means the shelf life of the fruit is declining. The peak bloom here is shorter than in itself. So usually you'll see bloom for a long time, but the peak bloom is about 2 to 3 weeks versus what we've seen in the South is maybe 4 to 6, 8 weeks. So it's more concentrated up here. And you see less of this overlap on the tree. Okay, so we've looked at flowering on the trees up here compared to Irvine, and you see less overlap of the male and the female flowers on the same tree, which would lead me to think that you definitely need to have you need pollinizers and pollinator varieties up here. I'm going to switch topics now. I think as you are purchasing trees, you need to be aware that you need to buy clean material. To my knowledge, we don't have avocado sunblotch up here in the San Joaquin Valley or in the Delta, and we want to keep it that way. Sunblotch is rampant down in San Diego County, and it's present in Ventura County. This is a viroid, and you can see what it does to the fruit. You can see what it does to the leaves. You can see what it does to the stems. And it is spread by pollen, grafting, pruning, infected seed. And I know from running the breeding program is also spreads through root grafting. Okay, so you want to ask when you buy trees from your nursery if they're indexing for this viroid. You want to make sure that you don't plant trees with sunblotch in your grove as much as possible. Okay, and fruit quality and yield can be affected, obviously. But the weird thing about this viroid is that the viroid will be active in the tree, but the tree could, quote, recover. But if that tree is stressed by water stress, heat stress, cold stress, it will express the viroid symptoms again. Okay, so I'm just urging you, as you buy trees, buy from, ask your nursery source, are they indexing their source material for sun blotch? So I was asked to talk about what is the difference between a seedling and a clonal rootstock. We've talked about clonal rootstocks here. So seedling rootstock is exactly what it sounds like. You plant a seed and you graft the sign variety on it, and you have a seedling rootstock tree. Those trees are usually very, very vigorous. It's a relatively short time from planting to grafting with the sign to the field planting. But you may see great variability between plants due to this phenomena we call high heterozygosity of avocados. Okay, so there's a lot. Avocados is considered to be only semi-domesticated. Unlike citrus, we don't have nucellar embryoni like citrus, where you have low variability between seedlings. You have high variability between seedlings. And you'll be able to see that when we go out, because we have a big planting of seedlings out in the field, and you'll be able to see this kind of variability. Okay, so that is the downside, potential downside of seedling rootstocks, is that you can get high variability from tree to tree. And that usually is very obvious when the trees are older, and you can see some big trees, little trees. Maybe some trees have iron chlorosis, other trees don't. Okay. Now, very common and a very good seedling rootstock is Zutano seed. Zutano is trees on Zutano seedling rootstock tend to be cold hardy. They tend to be able to tolerate heavy, wet soils better than a lot of other rootstocks. And so, and the trees are very vigorous and come into production relatively soon. Okay. Now, clonal rootstock is essentially a double grafted tree, and I'm going to show you some pictures of what that means. They take longer to produce, 18 to 24 months. You usually observe, because it's using the same genetic material for every tree for the rootstock, you usually see greater uniformity in the field. And the basis of going to clonal rootstocks was grounded in this search for Phytophthora-tolerant rootstocks, because Phytophthora cinnamoni is a lethal disease to an avocado tree. So this is what drove the development of clonal rootstocks. It was a search for something that we could use that was tolerant to Phytophthora. Okay, and common rootstocks is not inclusive: Duke 7, Toro Canyon, Duza, Sedum, Leola, Zarella, Tammy, and the list is growing and growing and growing now, because we're having more and more rootstocks, clonal rootstocks. So significant development in avocado rootstocks. We had the discovery and identification of Phytophthora cinnamoni in 1940, and the University of California hired George Zentmeyer. He was a forest pathologist, and he went down to Riverside, and he spent the next 40 years being an avocado root rot scientist. We have the development of the etiolation process by Ted Froelich. He was a researcher at UCLA when UCLA used to have horticultural research. But he developed a method that wasn't commercial. So we really, the world owes it to Hank Brokaw for coming up with a commercial method. And that method was slightly modified by Andre Ernst in the 1990s. The other thing is, in Israel, you have Avraham Benyakoff, who was a researcher in Israel. A big challenge there is not root rot, but with salinity. And so he was looking for rootstocks with salinity tolerance. And then from the 1990s onwards, now that we have clonal rootstocks, the effort has been, okay, besides root rot, what else are we going to look for? So I just want to give homage to George Zentmeyer. He was definitely a pioneer, and he worked with Gene Schieber, who was a coffee pathologist in Guatemala, who did a lot of foreign exploration for material in Central America looking for resistance. Here's a picture of Ted Froelich, and he is the reason why we have clonal rootstocks today, because he spent 25 years trying to figure this out. And this is actually a picture of the very first clonal rootstock trees. And this is from an article from the 1970 Avocado Society yearbook that is available on avocadosource.com. All the yearbooks are available on avocadosource.com. And here's a picture of Hank Brokaw, who figured out a way to commercialize what Ted Froelich. So he was able to take it from the ivory tower to practical use. And here's a picture of Abraham Benikoff, who is really a pioneer of salinity tolerance in rootstocks. And we have a suite of rootstocks from Israel now commercially available due to the work of this gentleman. The other thing that Abraham Benikoff is notable for is the fact of his crusade for germplasm conservation in the world. And really, that is now led by Alejandro Barrientos, who's from the University of Chapingo, because the Wild Center of Diversity for Avocados is under great threat from deforestation. So the diversity was led by Benikoff, and then that mantle was picked up by Alejandro Barrientos, and Anaki and I and many others are very, very active in trying to build the case that we need to conserve our wild avocado germplasm. So this is just a diagram, just so you can sort of imagine what the cloning is. And so you start off with the seed, you germinate the seed, and then when the seed is probably about this tall, you graft the rootstock on. Okay, so you may start with a Zutano seedling, and then when that seedling tree is maybe a foot tall, you go and you graft what the rootstock will be. Okay, and when that rootstock bud starts growing, when it's about this big, you put the whole thing in the dark. No light at all. And that bud then, as it grows, is growing in the dark, so it's very etiolated. And it'll be, you leave it in the dark at about 75 degrees or so, and the thing will be about this tall after about a month. You take that out of the dark, and you wound that etiolated stem, and you put a growth hormone on it, and you cover up that white stem. And what you see here then is in this situation, you start getting roots. Okay, so that is what Ted Froelich developed and what Hank Brokaw commercialized. And in this diagram, because such a nice diagram, this is the Ernst modification of that method, where eventually you cut off that tree, that the rooted cutting from the nurse seed. And then you put the sign on. So that's when I said it was a double grafted tree. That's what makes a double grafted tree. So what about the future? I think seedling rootstocks are fine if you don't have root rot. It's fine if you're saying, I'm going to rely on phos acid, I'm going to rely on arindus, I'm going to rely on mulching. We have a lot of trees in Southern California that are still old trees that are still on seedling rootstock. So you can use seedling rootstock. That's fine. Tissue culture, that's a new frontier. We've had tissue culture trees sold here. We have tissue culture trees being sold in Australia. A lot of activity here. And probably within the next, I would think 3 to 5, maybe 8 years, maybe we will be replacing the Brokaw-Ernst method of tree production and going to tissue culture. I don't know, but the crystal ball says, it's becoming more in focus. So with that, I'll say thank you. And now for the next, if I could have the speakers come up, and our known is still on. If now we can just open it up for free-form questions. If anybody have questions about any of the talk, this is your chance to ask anything you want. Yes.
For the overhead cooling, I think in his research, he was talking about around fruit set and fruitlet. Is there a time of year or in the season that the overhead cooling is, you get more bang for your buck? It's hot here all summer long, so should you be running it all summer, or is there a point where they're set and you're not gonna get the drop and you're not gonna get a benefit to that? Or
So Arnon, did you hear the question? The question is, the data you showed was really around overhead cooling during flowering, but it's hot here in California, from now to September. So is there a more important time versus a less important time to have this going, or would you be looking at it all summer?
Just to emphasize, we did not operate it during flowering. We operated it directly after flowering. There was a question earlier about the impact of overhead cooling during flowering time. As Inaki said, this is kind of question mark. I'm not sure what is happening. So we apply the overhead cooling when we have a young small fruitlet. Now remember, the heat wave that we are speaking about is combination not only of high temperature, but also of low humidity. If you have relatively high temperature with high humidity, it's okay for avocado. The stress is not so hard, but if there is combined stress of high temperature with low humidity, then the stress is very strong. I propose some threshold, and you might see whether you stand on those threshold or not. We don't have some evidence about the impact of later on operation of the system. A question that you need to ask yourself, if you have damage, for example, if you have damage for the fruit quality because of sunburn or other aspect during later development stage of the fruit, you might consider using it if you have a severe heat wave, but I do not have direct answer for this.
You want to say something, Ben?
Yeah, the two events that have happened in Ventura were episodic. Once in 2020 and another time in 2022. And the systems were run late summer, early fall, and it was to retain fruit that was going to be harvested not too distant future, the next week or so. And there was significant fruit drop in orchards that didn't have the cooling, and these two orchards that had it retained their fruit. My experience prior to that was with misting systems down in San Diego. George, I forget his name, down in Fallbrook, had a misting system on his avocado tree. And
George Emmerich?
George Emmerich, thank you. Was running it constantly when it got 90, 95 degrees, and the tree's completely defoliated. So this, thing about the salts is real. It gets on the leaves and it's absorbed by the leaves. And as Arnon said, it's, you really have to have good quality water if you're going to run it for very long.
So I'll add, Jose Avina's in the room, so he knows what I'm talking about. There used to be a grower over here by where, Olive Hill. And he put misters on his wind machine. And he would run it when it got to like 100 degrees. And I went out with Jose, I believe it was you and a couple other people, and you were under where the area where it got wet with the wind machine. The fruit were noticeably larger than as you walked away from the wind machine. So, that was my first experience. It was like 20 years ago. That here was something, a guy just decided he was going to do that, and it worked.
So your cooling, you want to start between 90 and 95?
The avocado, as a general rule, depending on the relative humidity, the stomates will start closing right around 90 to 95 degrees. If you have lower humidity, they close sooner. Higher humidity, it'll delay it a little bit.
I've got a question for Inaki. What about supplemental pollen? Putting out pollen from another source, sprinkling it around?
Right to the wind machine.
In which way, Ben? Like using
how does it work? Does it work?
Well, we don't know if it works. We have a project.
Yeah, we have a
Yeah.
project.
So we'll give you some results, I think, probably next year. As I said, it's tricky because the avocado pollen has a low viability period. So it's very difficult to conserve the pollen and then apply, and that the pollen is going to be viable. There are some companies that are selling, as you know, pollen for pollination. And there are two things there. First, that the pollen has to be viable. And we're going to check that. And also the pollen has to reach the stigma of the flowers. So you have to release the pollen, and you know that the stigma of the flowers is very small. So you have to have a mechanism in which you can make the pollen available to reach the stigma of the flower. And the avocado flowers, also the amount of pollen that they produce is very low compared to other crops. Like for example, the walnut. Of course, you have tons of walnut pollen that you can apply with a blower. That's not the case in avocado. So we have this project. We are studying different systems and see if it really works in improving fruit set.
Yeah, so I should add, since our sponsor is here, it's funded by the California Avocado Commission. And Rachel's one of our cooperators, actually. And so we're using, we have the control, which is no supplemental pollen. There's a dry pollen that's being sold, so we're using that. And we're applying it with the in the grows we're doing it by backpack sprayer. That pollen has a compound called SureSet in it, which is a bunch of stuff in it that I don't know. And then we're working with a Chilean company, Biopollen. Their pollen is suspended in a liquid formulation. They are applying the pollen because I don't want anybody to say that if we get bad results that I applied it wrong. And then this year, we're going to be working with Bloomex, and Bloomex is a company from Israel. They have a different approach. They're using an electrostatic, the fact that there is an electrostatic charge difference between the pollen grain and the sigma. And so we're waiting to get that equipment. It's going to come from Mexico, I found out yesterday, because all flights are canceled from Tel Aviv. So they can't ship the equipment, but it's coming up from Mexico. And we're going to have that as a third treatment this year. And the other nice thing in this project is because of Bloomex, we're going to be also looking at the influence of different pollen sources on fruit set. That'll be done with Hass because we're doing the project down in Ventura. But so that's the project. It's going to go for another year and a half approximately.
You said pollen sources. What do you mean with that? Is a different type of pollen than avocado? Is a different color?
No, so the question is about different, we're using different A pollen because right now for the project, we're using Zitano pollen or bacon pollen, depending on the grower, for pollination on either Hass or Gem. But the question that was raised in Israel, and it's a good question here, can you use Hass, can you artificially add Hass pollen onto Hass? Can you add Gem pollen? Could you interplant Gem and Hass or lamb Hass and Hass? So we're going to take different A flower type pollen and apply that.
Jackie, you can study on that and say that it's different between varieties.
Yes, the question is if there are differences between varieties in pollen quality, and that's true. We did an experiment some years ago, both in vitro and in vivo, and there are some varieties that produce pollen with a higher quality, higher viability. In fact, when you mix pollen from different varieties, there are some that are more successful in fertilizing the ovule. But that's something that we have to expand the knowledge and see if we can decide which variety is going to be the best for each particular environment, because some varieties the pollen is going to behave better at higher temperatures and some are going to behave better at lower temperatures. So maybe you have to you have a different pollinator tree depending on your environmental conditions. There is not like a universal pollinator for every condition.
I'd like to emphasize that avocados are still a wild beast. And this concept of pollination has been going on. If you look at the avocado yearbook back to 1924, pollination, how many hives and how many, it's my predecessors, George Goodall in Santa Barbara and Bud Lee and Ventura fought over it. George said, you don't need anything but Hass, and Bud said you have to have 15% bacon, zutana, whatever out there. It's just,
It's very controversial.
yeah. But there's finally going to be some good information. Right.
In this project, the question is, are we using insects? In this project, we're letting all the growers are using bees.
Oh, so that's an issue.
Yeah. No.
Is it just a few species of flies that can pollinate, or most flies will do it?
We've seen different species of flies. The thing is that I don't know here in California, but in Spain at least there are some species of hoverflies that you can buy them commercially because they are hoverflies, like the
Syrphid flies.
flies.
Oh, okay.
And you can buy them commercially because they are used for pollination in different crops in the greenhouse. So you can buy the flies.
But what about a house?
House flies, you can find them also in the orchards, and they can pollinate the avocados. But we don't have information about how good they are compared to other flies. So we are using the commercial ones because that's the ones that are available. But there are a wide range of different flies that can be present in the avocado orchards.
There was a study done in Peru where they have a lot of chicken farms in Peru, and so they noticed that they had higher fruit set closer to where the chickens were. So there was an experiment that was done where they hung meat in the trees to attract the flies, and they got higher fruit set.
What about chicken shit?
But Ben's been working on this topic as well.
Yeah. And in Australia, they put bags of raw meat and throw it to the orchard, and that attracts flies, and they say it works.
Yeah, that's the main problem with flies. I mean, with honeybees, the bees will be back because they have to go to the hives. With flies, you release the flies and they can go anywhere. So you have to develop some strategies to maintain your flies in your orchard.
Can I add something to this discussion?
Sure.
We are looking on the effect of flowering cover crop on pollinators here in Israel. And one of the interesting findings is that when you seeded cereals, you increase the hoverfly population. I didn't know it earlier, but the cereals are kind of host for aphids. And the aphids secrete something that the larvae of the hoverfly eat. So when you have cereal in the avocado orchard or nearby, you increase the overfly population, and the overfly are very important. I think after honeybee, overfly, as Sinaki said, are the second important pollinator of avocado.
Yeah, at a trial that we have in Ventura, and again, Ventura is not Porterville. The most common visitor of avocado flowers is hoverflies, syrphid flies, but they don't carry as much pollen as a bee would.
But what are they after? What's the plan?
They're going after nectar and pollen. They're feeding themselves. And an avocado flower, I mean, you look at the avocado flower, it's just a huge resource. There's nectar and there's this pollen, and there's all kinds of things, crawling all over it. Hummingbirds love them. So,
Craig?
So one thought on your cooling where you have the residual salt, I assume your bicarbonate level in your water was relatively low. So have you thought about running a wetting agent in there so that when the water hits the leaves, you don't have much retention on the leaf, and basically majority of that's going to run off because you've broken the surface tension?
that's a good, we haven't done that.
Repeat the question.
Oh, the question is running a surfactant with the water as a cooling agent. Actually, but you want the water on the leaf. You don't want it to run off. But it would run off carrying the bicarbonates, yeah.
Well, I don't think if you detected your sodium chloride, then you don't have much bicarbonate, because then the calcium is going to basically attach to the bicarbonate. It's going to give you creline. But if you're able to wash it off, then that's really a salt, not a lime deposit. But I was thinking, you're shooting for evaporation, but if you decrease the surface tension, then you should have not so much residual water left on the leaf to keep from having that absorption. We don't have a lot of high salt water here to begin with, not like you guys out there. But anyway, that's a thought.
Can I respond to this?
Sure.
It's very good comment, and we look on this aspect from different angle. When you look on the leaf after you apply the water, you see droplet. The waxy surface of the leaf allow only droplet to be accumulated. It's not cover all the leaf. And then you might think that the effect of evaporation is lower. So we try to use surfactants, and it really allows the water to spread all over the leaves. But when we look on the effect on the temperature reduction, we didn't saw any impact. As this person said, I think we need to explore more on the issue of salt accumulation. It's interesting approach.
Any more? Oh, yes?
Mulchy question, are softwoods preferred? I mean, do you want a faster breakdown than like hardwood?
You want
Repeat the question.
what type of material should you use for mulch? And it's whatever you can get your hands on that's coarse, textured, relatively large. Softwood, yeah, I would presume, bark is the ideal one, but you can't afford to use it.
I would say that I have a research plot in Ventura County that they took out lemons and they shredded the lemons in place. And I can tell you that has made a wonderful mulch. The trees are 12 years old and we still have the shredded lemons in there. And we have wonderful root growth up into that interface with the shredded citrus. Any more? Yes.
I have a question for you regarding the mulch. The valley growers are putting out by the hundreds acres of walnuts. Is the walnut, what it gives off toxic to the avocado roots?
That is a great question. Is walnut wood toxic? And this is a study that we did 30 years ago. We looked at oleander, eucalyptus. We didn't do walnut, but the idea is that there's these anti-growth inhibitors within some plants. If you go into a walnut orchard, there's lots of weeds. There's not supposed to be any. And I would say any woody material is fair game as a mulch.
Thank you.
So allelopathy it's in textbooks, but I don't think it's in reality.